The HiT platform enables us to generate T-cell receptors, or TCRs, that instead of binding to peptide HLA complexes, they bind directly to cell surface proteins. As these TCRs are HLA independent in terms of binding antigen, we've called them HiTs. And we're really excited about HiTs because this technology opens a whole new range of targets for us that have to date been addressed by CAR-Ts or other cell therapies that use antibody moieties to engage with the antigen. And this enables us to target solid and hematological cancers without the need to select patients based on HLA type, so we can treat a much wider patient population. We believe that HiTs have some potential advantages over CAR-T cells and TRUCs, both of which use high affinity antibody moieties for targeting the antigen. For example, HiTs have normal TCR signaling on contact with antigen, unlike CAR-Ts, which have been associated with tonic signaling, and this can lead to increased incidence and severity of adverse events like CRS and neurotoxicity in the clinic. In addition, many molecules, including mesothelin, are cleaved from the cell surface, releasing soluble fragments that can act as a sink for CAR-T cells and TRUCs, inhibiting their function. We've been able to show that soluble mesothelin does not prevent the function of our HiT T cells, as they have lower affinity for antigen, but high functional avidity. Excitingly, our mesothelin HiT induced complete regression of pre-established tumours in a mouse xenograft model. And importantly, it showed much greater efficacy than a TRUC construct in the same model, which demonstrated only a minor reduction in tumour growth rate. We announced in November last year at our Investor Day our 2252 plan, with a commitment from research to bring 5 new products to the clinic in 5 years. Towards this goal, we're building multiple opportunities to maximise the value potential of our current SPEAR T-cell products and to expand our pipeline to include HLA-independent TCRs and next-generation TILs. The announcement of the first preclinical data from our new HiT platform marks a significant milestone towards achieving these objectives, and the work we've done with this first candidate establishes a preclinical testing strategy to evaluate HiTs against additional targets, including GPC3 for liver cancer. We'll be giving further updates on our pipeline in due course. The announcement of the first preclinical data from our new HiT platform marks a significant milestone towards achieving these objectives. To maximise the potential of our SPEAR T-cell products in the clinic, we're developing TCRs to additional HLAs. We announced a new partnership with Inger Maria Svena and her team at the CCIT in Denmark. In this collaboration, we aim to develop next-generation supercharged tumour-infiltrating lymphocytes, or TILs, to improve efficacy in patients with melanoma. And the first product we intend to take to the clinic in 2022 will be a next-generation IL-7 TIL. We've also built a toolbox of next-generation T-cell products. Our first next-generation product combines MAGE-A4, targeted SPEAR T-cells, with a CD8 alpha co-receptor, and this is giving promising clinical data in our SURPASS trial. And with Neuroimmune, we're exploring the combination of IL-7 and CCL19. And these are just examples of some of the next generation approaches we are taking, and we're very much looking forward to taking the best of those into the clinic.